In this example, we will analyze variants in mitochondrial genome, which is a non-diploid mixture of actually multiple genomes, and so this is also applicable to the analysis of variation in viral, bacterial, and other organellar genomes. In all those cases where you expect continuous allele frequency from anywhere between 0 and 1 or 0 or 100%. What I have here is deduplicated clean BAM file, and the first thing I will do, I will downsample it using Picard downsample SAM tool. This will allow me to get its size down, so all downstream analysis I will be performing will be faster, but they will still be representative of the larger data set. And once, for example, if you're experimenting with this pipeline when you're doing it for the first time, it's always a good idea to downsample, because once you are sure in the choices of your parameters, you can then rerun the rerun the same thing on the entire data set. So I will move this slider to about 10% and click execute. After downsampling, I have file which is about 10% in the size of the original dataset. So now let's go and call variants. For this, we will go to NGS variant analysis. And there are two tools which I can use for continuous examples, continuous allele frequency examples like this with non-diploid scenarios. One is called naive variant caller. I will use it in this screencast. And another one is called freebase. I will use it in the next screencast. Let's click on naive variant caller. And you need to tell what dataset you would like to call variants, in which dataset you would like to call variants. This is in this case dataset number 2. And what's the reference genome? In this case, the reads were mapped against HG38 version of the human genome, so we're going to choose it. I will restrict this analysis to mitochondria only to make it faster, because we're really interested in only mitochondrial calls here. I will require at least 100x coverage for a site to be considered, with a minimal quality of 30 on a Fred scale and a minimal mapping quality of 20. So site needs to be covered by at least 100 reads. In each of those reads, the base corresponding to that site should have quality of at least 30, and the read mapping quality should be above 20. It's a mixture, so it's definitely not diploid. We'll set ploidy to 1, and we'll set to report counts by strand. This will help us down the line to see if we have any strand biases for the calls that we will find. Now naive variant caller has finished, and it reports data in a VCF format, which is not very pretty to look at, and it reports data for each site regardless of whether you have alternative alleles or not. So before we do anything further, let's filter this data set using NGS VCF manipulation VCF filter. And we will filter it so we'll only retain those sites where alternative allele frequency is above, say, 5%. And this restricts our dataset to just 31 sites. Again, this is not something you really want to look at, so what we can do is we can visualize these data in genome browsers, such as, for example, integrated genome viewer. But before we do that, we need to make sure that Galaxy knows which genome this data set is derived from. And if there is a question mark here, that means Galaxy actually doesn't know about this, but we know that the reads were mapped to HG38, so let's set this to HG38. So if we click on this question mark and set HG38 and set it to HG38 here, you will see that once you save and the history refreshes, you will be able to display in actually several browsers such as UCSC or IGV using human HG38 data. So what I'm going to do, I'm going to click on this link, and eventually it would download an IGV, and ultimately you will see IGV opening on your desktop. It shows all chromosomes at once. We only care about mitochondria, so I'm going to switch to mitochondria here. And I will zoom into one particular region here, because as you can see, there's a cluster of a number of variants here. This site is very interesting because It's one of those rare cases where you have complete switch during mitochondrial transmission from mother to her child. At this position, A is the reference base, alternative allele is G, and this is what you see in mother, these two gray boxes in mother. A is the major allele in both blood and cheek. But once you look at the child, you will see that it changes, it flips completely to a G. G now becomes the major allele. And the problem is that the particular change at that site from A to G is known to be linked to a number of severe mitochondrial disorders, such as, for example, mitochondrial encephalomyopathy. So we don't know what's the status of that child. In this case, samples were taken from normal people, from normal healthy individuals. Of course, when you're doing this analysis for the first time, you might not know about any sites like this. So let me show you two last steps in the workflow for the variant analysis using a variant caller. We're going to use one more tool. It's found in variant analysis section again. It's called the variant annotator. This tool works on the output of naive variant caller, and it tabulates a number of statistics, number of parameters, which are very useful for finding, for example, sites that, like the one that I just showed you in the genome browser. So I open this tool, and in this you can set what's the minimal allele frequency you want to look at. Let's set it to 1%. Let's set the coverage to 100. I would like to see output by strands, so we can again be able to detect any strand bias in our calls. And let's write the header line. I will click on execute. You will see that the output of this file is just a tab-delimited data set. Let's look again at our site 3243. You can see that it outputs line per sample. We have 4 samples in our VCF file. And it tabulates the number of A, C, G's, and T's per strand in each of the samples. And in the end, it tells us what's the major allele, in this case A in the mother, G in the child. What's the alternative allele? G in the mother, A in the child, and the frequencies of minor allele. So you can see here that the minor allele in mother is on frequencies ranging from about 15% to 30%, in the child from 40 to 30%, but these are different minor alleles. In mother it's G, in child it's an A. And in this format, it's relatively easy to find sites like this simply by filtering on the last column here, column 16. So I can go ahead and use filter tool to filter all the sites from this data set where column 16 has non-zero values. I have one header line in this data set. And so now you can see again that our site 3243 is featured here prominently. So this is an example workflow on how to call continuous frequency variants in non-diploid